So we're back in E443, 543. It's our fourth lecture. Today we're going to be talking about writing computer simulations in Python, how to use Twedit effectively, and how to plot things for analysis. And I hope we'll get to cell growth and division as well. And since the class is pretty small and people are fairly experienced, I think I'm going to try going a little bit faster today than I have in the previous lectures. And as always, I need to remind people this is live streamed and recorded, and a couple of people said they were sick and couldn't watch it, so they said they needed the recordings. So I don't know if people have had any chance to think about project ideas. Maybe with so many people not here, it's not the best time to do that. So maybe we should hold that discussion for next week. But I would like people to think about projects. And I would encourage people, especially people who are less experienced in modeling, to pair up. And so if you're somebody who has not done this a lot, and try to find somebody who's got more experience and partner with them. Otherwise, these projects can be they don't have to be very time consuming. They have a tendency to be time consuming. And having two people work on a project rather than one often works better. Okay. And then we're going to review CompuCell program model specification, architecture, Twedit, and then we're going to do some exercise. And we have, I hope, getting to both cell division and plots. So I don't know if anybody wants to bring up some project ideas now, or whether people feel that we should wait for next week on that. Does anybody have something they want to say now about it?
So my undergraduate background is in math and cognitive science, except unfortunately, I didn't do much applied math in college. My present day background is in doing like mathematical and computational modeling in neuroscience, specifically having to do with navigation. There are these weird cell types, specifically like place and grid cells that have seemingly been implicated in rats' ability to navigate. And the cells, I guess what I find so weird about them is they have these firing fields that actually like tile space. They make this really interesting hexagonal grid pattern. I'm thinking about grid cells specifically. And that's already reminded me of a lot of things we've seen in the course. So I was thinking of doing a project that attempts to get at how these cells might develop. Like if you say that there is, say, random firing in some like young neural population with some simple learning rule. And like maybe the project would look like, okay, formulating a handful of different possible learning rules and then testing them with the same kind of random firing in a population and see if we get like grid-like firing. But I guess you would have to see the cells that I'm talking about, see their firing fields for that to make sense. But if anybody else is interested in that, that is something that I'm quite willing to do. And then the other thing that I would consider doing if I were talked into doing something that's not going to fit into the rest of my research is I'm interested in whether like blood vessel tissue dysfunction, I don't have like any medical background, so I don't really know what I'm talking about, is implicated in some cases of long COVID. Because I've just read that and I had long COVID like a year ago and spent like so much time trying to fix it. And that was one of the things I kept reading about. Yeah, so if that's something someone else is interested in, we could also talk about that. Yeah, that's my stuff.
They're both interesting problems. The long COVID problem is quite interesting because, as you say, it seems to involve, at least in some cases, vascular disruption. One theory that has been put forward by Tanover at NYU, which I think is quite interesting, is that a lot of long COVID happens because of microglial overactivation. And so if you look at the typical pattern of immune response in the body, you'll have early activators of inflammation like IL-2, IL-6. And then later on, you'll get things like IL-10 and IL-12 that come in and try to shut down or at least modulate the level of immune activity. And so, uh, and so then you have, you can ask the question, what's happening to shut things down or not shut things down? And there's some nice ODE models, simple models that Bart Ehrmantraut did with some undergrads a year or two ago, where those models have, and actually Julia Arciero's published some models of that kind too up in Indianapolis, where you have models of the early immune response, the early innate immune, the later innate immune response, and the adaptive immune response, and then the shutdown. And it's possible to build models where you don't get enough immune response, the virus runs away and kills you. You can build models where the feedback, the pro-inflammatory feedback is so big that you get essentially cytokine storm and then the immune response kills you. You can have situations where things work, where immune response kicks in, clears the virus, and then turns off again, which is what you want. And then there are also ones where you get kicked into a chronic inflammatory state, where you get rid of the virus, but the inflammation never turns off all the way. And it winds up that in the brain, there must be some kind of shutdown signals for the immune response, but they're not well understood, and they seem to be much weaker. And so one theory was that in the brain, when you have microglial activation, where microglia are one of the immune types which are a little different in the brain from elsewhere, they are causing tissue damage, and the damaged tissue itself is sending a pro-inflammatory signal that keeps them activated. And so basically, the virus triggers something which is a self-maintaining damage mechanism. And this was one of the reasons that he believed this was the fact that the anosmia, the disruption of smell that you get, as people who've had COVID, and I probably, unfortunately, almost all of us have at this point, and also somebody who was, I wasn't fortunate, maybe I wouldn't call myself having had long COVID, but I was more or less confined to bed for 3 months after I got COVID the first time. It took me quite a while. Well, 3 months is sort of medium COVID, I would say. Uh, but the smell disruption actually is quite interesting from the point of view of time scale, because if your disruption of smell where we're killing sensory receptors, then you would expect recovery to be pretty fast. If the sensory disruption where we're killing the neurofibers to the brain that are carrying the olfactory information, you expect never to recover at all. But instead, you get these recoveries that are very idiosyncratic and typically 6 months to a year in length. And it was almost impossible to come up with a mechanism that wouldn't either be much faster or much slower than what they see. And so it seems that in this case that the olfactory receptors under stress can go into some sort of protective state where they don't signal properly, but they don't die either. If they died, there would be proliferation and replacement. But they're there but non-functional and getting in the way. And again, it was a self-sustaining loop of feedback. In other words, even though the virus was gone, the damage signal itself was causing the other receptors to stay in the damage signal state. And I'm not an expert. Emma, you're welcome to ask a question, but I don't know that I'll be able to answer it. But to me, this was quite an interesting set of insights.
Is it possible to do some sort of imaging in humans to see what's going on with the receptors? like whether they're present or dysregulated or?
So Emma asks, is it possible to image the nasal passages to look for receptor damage? I don't know if it was done in humans. Certainly in rodents, you can do it.
Or you could just.
Well, but you want to be able to do it longitudinally. You don't want to have to kill the animal to be able to make the measurement. And so I don't know what the markers are for this refractory state, but certainly there is some cell death when you get SARS-CoV-2 infection in the nasal cavities. But it's not that pervasive. The amount of cell death, the fraction of cells infected is not as high as you would expect. If I remember right, this is going back to 2020 when they were first publishing on this. The fraction of cells that are infected is not that high, and the size of the lesions is not that big. You're not getting a lot of pervasive cell death. It's a bit different in the lungs, where if you have serious consequences from the lung infection, you can get big lesions and pervasive ones. In the nasal passages, it wasn't that much. And again, that made the disruption of smell even more mysterious. If you killed all the receptors, well, you'd understand why you couldn't smell. But the receptors were there; they seemed okay, but they weren't transducing the signals that they were expected to do. I apologize for not having kept up on that literature in the past year or so, but Tanover actually wanted to do some modeling of COVID. He felt, and this is something that you see a lot now that there are these antivirals, people who take, well, nobody uses remdesivir anymore, but Paxlovid. Paxlovid gets rid of the virus quite effectively, but then people have rebound. And then with the rebound, they don't clear it. And one theory of this is that what's happening is that your adaptive immune system effectively sees the decrease in viral load, says, okay, the innate immune system is taking care of this. I'm not going to kick in the adaptive immune response, which would then give long-term protection. And there are these adaptational behaviors in non-self recognition, of course, is fundamental to the immune response. One of the sort of general vague ideas in immunology is that things that happen over long times get learned to be self. And so the idea here with long COVID may be that the contribution of specifically of a drug like Paxlovid to this problem is that by stretching out the clearance of the virus, your adaptive immune system basically says, oh, well, the virus is here. It's not doing any harm. Therefore, I'm going to tolerate it rather than get rid of it. Now, that's, of course, very anthropomorphic, and I don't mean it to be taken too literally. But one can imagine them building a computational model, maybe an OD model, maybe an agent-based model, where if something is present for a long time, the production of CD8 plus T cells and related B cells, memory T cells, gets downregulated. Tregs get put up for that. And so that the immune system basically then says it's going to tolerate the infection. And this isn't quite as crazy as it might sound because, of course, bats, which are the source of a lot of these viruses, have the same immune components as that we do, but they handle immune response very differently. They have much stronger innate immune responses and much weaker adaptive immune responses. And so bats typically don't clear viruses, they live with them. So the bat will get infected, won't get very sick, it'll keep the virus under control, but doesn't get rid of it.
Could you repeat the terms for the two immune responses?
So innate immune response, and the distinction between innate and adaptive immune response isn't quite as useful as it could be because there's cellular innate immune response and early innate immune response. antiviral response. And there's also a cellular adaptive immune response, and also things like antibodies, free antibodies, which are non-cellular adaptive immune response. So both innate and adaptive immune response have cellular and non-cellular components. And the sequence in which they happen is sort of alternating. But fundamentally, innate immune response are the responses your cells and your tissues have to a virus or a bacterium that it's never seen before. And then the adaptive immune response is when your body has developed specific antibodies that recognize that particular pathogen. And then you have eventually store a memory of those particular pathogens so that the next time you are insulted by them, you can mount a much stronger and faster response. And so then there's a distinction between the primary infection the first time you're exposed to something and then repeated challenge. So, okay.
And bats have strong innate and weaker adaptive, or is it a little bit?
So bats have strong innate immune response and weak adaptive immune response. There are a whole series of molecular species, and I try to, in this class, avoid long lists of gene names. But the molecules that regulate the immune system are called cytokines, and they come in lots of families, which typically are something number, which is not always easy to remember which is which. But the interferons are associated with very early response. So when you get a virus infecting a cell, typically and this is what Pedro's working on at the moment. This virus, the cell will send out an interferon signal that tells neighboring cells there's a virus around, be careful. And it's not exactly clear how cells are able to resist viral infection after exposure to interferons, but they enter what's considered to be an antiviral resistant state, in which they either don't internalize the virus, or if they internalize it, they don't then have the virus proliferate inside of them. And one of the things the viruses have to do is suppress that response to the interferons. And if they don't do that, then the virus will enter the cells but never cause them serious infection. And that all happens over a period of about 4 to 6 hours after first exposure. Now, in bats, interferon is turned on all the time. And so the bats are always effectively in a state as if they're primed to watch for viral infections. There are a lot of other theories about what's happening in bats, but that's certainly one of them. In humans, if you have interferon turned on all the time, then you get tissue damage. So it's not something that we could do. You might be able to do it prophylactically, and they tried it, I think, early on with IL-7 they used as an inhaler. But the viral, as I say, the bats have all the components of the immune system are the same. Bats are mammals, but the way they're synchronized and organized is a bit different, and the emphases are a bit different. All right, boy. Okay. Anybody else want to talk about projects, or sounds like Pedro, maybe you and Emma should talk a little bit about immune response. Maybe there's I mean, I think the idea of doing patterning and columnar activation could be interesting too. You could take that delta notch type model and ask the question, could that be generalized to things you're interested in? But so you might want to talk and explore. Does anybody else have something they wanted to talk about now?
Yes, I can talk about my.
Okay, go ahead, Pedro.
Hello. So my idea is developing a tissue in cell with the mechanisms that James was describing. And so it's basically a tissue, and there is a virus replicating in each cell. The virus triggers production and release of interference. In general, the early cells infected, they will die, but the surrounding cells, by detecting the high levels of interferon, can develop an antiviral state. And the virus has the capacity to shut down these pathways. So they antagonize each other. Cell innate response antagonizes with virus mechanisms that try to shut down the cell defense mechanisms. So this is as simple as it can get. Unfortunately, when you look up in the literature, there's lots of pathways. So it can be as complicated as we can, but that's not the idea. The idea is to try to simplify and abstract the main mechanisms. So if Emma is interested in working with this, can definitely join on this project. That's it.
Okay, anyone else? I mean, at this point, I don't think we need to be going into great detail, but I think it is important if we're going to have people team up to at least have some idea of what people's interests are. And so I would appreciate it if people would talk to each other. You have each other's email addresses, and I think I'm going to ask people for next week maybe to write a half page. Doesn't have to be very detailed, but and it could even be 2 ideas or 3 ideas. Just a half page where you outline a couple of ideas of projects, and then we can circulate those and see if there's some commonality. Would that be a reasonable way to do it? In the past, I've created a Google Doc where people could fill things in, but then they tend not to do it. So I'm going to make it an assignment. So that might be okay. And we're missing some people today, so it reduces our ability to have the conversation. And if people don't have an idea of a project, there are lots of simple things that we need done, or it would be nice to do, that it would be good learning projects. So feel free, if nothing, if they sound too complicated or not interesting to you, and you want a basic project that would be appropriate to learn from, I have plenty of suggestions. So I want to come back. Last time we talked about fluctuations, reasonable behaviors. We went over that. We talked about the idea of acceptance and how changing the fluctuation scale changes. If we change the fluctuation scale and the energy scale by the same factor, we don't change anything. If we multiply our energies by 10 and we multiply our fluctuation amplitude by 10, the model doesn't change. It's the ratio of fluctuations to energy in the system that matters. And then we did some analysis of different patterns, both for single cell types and also for cell sorting, which you continued in the homework. So now I'm going to talk about CompuCell code a bit. Having had this long philosophical discussion and then the long biological discussion, we're now going to jump into some computing. So far, all of the simulations that we've done in the homeworks have been using the markup language, the CC3D ML markup language. And that's fine as far as it goes. And we've talked about extending it over the years. And people like Paul Macklin and the group in the Morpheus group in Dresden have built more elaborate markup languages that allow you to specify models, and there are a lot of real advantages to doing things that way. But CompuCell is designed so that a lot of the guts of the simulation, especially the data analysis portions of the simulation and the time-varying portions of the simulation, are done in Python. And so again, you've already used CompuCell, you have Twedit, the editor and CC3D player. And we looked very briefly the other day at what the components of a basic typical model are. There's a CC3D ML script, which we've looked at a little bit, and you've edited in the class. There's something called the main Python script, which in general we're not going to do much with. We'll try to avoid having to do anything with that by hand. Usually CompuCell will deal with it automatically for us. And then there's the Steppable script where you actually put in the user-defined things that you care about. And that's CompuCell Steppables are really the place where you build the complex models. So I want to talk about a couple of things that we could use. There are going to always be some people, especially if you're going to put your projects in GitHub, who really like to use an IDE like VS Code. And if you were going to build CompuCell today, you might use VS Code as an editor and build all the bells and whistles that we've put into Twedit into VS Code. Unfortunately, VS Code didn't exist when CompuCell was written, and so we don't have that. So Twedit, as a model editor, has some features that are really very helpful, especially when you're learning, but even once you've learned, which is that Twedit has what are called code snippets. It has templates. Occasionally there's one that's missing because somebody's new or because somebody forgot to put it in. And once in a very long while, there's a typo in one of the code snippets. But in general, Twedit has code snippets that mean that most of the things that you want to do are templated. That doesn't mean that the code that you get from the templates is exactly what you need, but you don't have to remember the names of functions. You don't have to remember the sequence of calling things because the templates will handle that for you. And one of the things that's puzzled me over the years when people learn CompuCell is how many people refuse to use the code snippets. And I really encourage you to do that because it reduces the error rate in coding enormously. So if you go into Twedit, you will see that there in the menu structure, and the order of the menu pull-down menus is different in Macs and PCs. But you will see two headings in the pull-down menus. One of them called CC3D. There's one called CC3D C++ for people who are really energetic and want to actually extend core CompuCell. You can actually do core CompuCell now. We're not going to cover that in this class, although Pedro can show you how to do it if you're interested. And then there'll be one called CC3D ML, which is for editing the markup language portion of your model spec, and one called CC3D Python, which is for editing the Python portion of your model spec. And so if you go and look at the CC3D ML one, maybe it's worth doing that together, and you pull it down, it'll give you a bunch of options: metadata, plugins, pots, steppables. And perhaps one thing about these pull-down menus is that the names and the order of things may not be the most transparent. We're not quite as good at Microsoft as Microsoft is at hiding the most important thing 10 menus down in an obscure place. But we try. So one thing that you do have to look at when you're going to use the code snippets is that you have to make sure that you put your cursor, the code snippet will get pasted in where your cursor is. So especially in the XML, make sure that you don't put your cursor by mistake in the middle of a code block, because the editor will happily paste the code into the middle of a code block and break the XML. But otherwise, it's not so hard to do. So if you go into an existing Python CompuCell simulation, go into the XML, find something that's not inside of a tag pair, and pull down the code snippet block there, CC3DML. You have a bunch of choices. And I'll just walk you through a variety of the things. You've already used Wizard to create some simulations. And I'll walk you through some of the things that you've got. If you open up the metadata block, it'll give you some options, change the debug output frequency, enable parallel execution, and then simulation properties. Some of these we've covered before. CompuCell is intrinsically multi-threaded. It's intrinsically parallel for multi-core machines. It won't work. It doesn't use MPI for multi-box machines, but it does work. It does use OpenMP for multi-core machines. And it will give you an option if you are inside of a simulation that's already there. It'll often say here, metadata module already defined, and then give you the option of either getting rid of it completely and starting from scratch, or commenting out what you've got and adding a new one. Certainly, commenting out and adding a new one is safer, so that you have what you had before. Twedit will generally try to populate the block with the parameter values that you already had. It usually gets it right, not always. Typically, you will have to do some hand editing of the code afterwards. Sometimes the code snippets will contain multiple options, and so they'll crash if you run them as written, because you'll have to pick one. One that we'll use more is plugins. These are those energy functionals that we've talked about. And if you pull down the plugins, you'll see some ones that are pretty familiar to us. You'll see cell type. You'll see center of mass, which we've always had ticked automatically. These are the things that are mostly in that checkbox list at the end of Wizard. There is surface at the very bottom, surface and volume constraints, which we're familiar with. There's contact energy constraint. There's a contact energy plugin. There's the cell length constraint that we've already played with. And then there are a variety of other ones that we haven't talked about yet. Some of which we'll definitely use in the class, a few of which are not so commonly used. There's the POTS model, basically sets those things in that opening screen of Wizard. How big is the lattice? What kind of lattice is it? How long does the simulation run? What's that fluctuation amplitude parameter? There's also a block called Steppables. And the difference between a plugin and a steppable is that the plugins are associated with the microsteps that are moving the cells around in the simulation lattice. The steppables are things that are called in between the movement steps in the simulation lattice. And they could include things like, which we've already used, like blob initializer. There's a PIF dumper and PIF initializer, which essentially are ways of reading and writing initial configurations. There are some differential equation solvers, diffusion solvers. And the newest version should have some additional ones for initial conditions as well. Okay. So that's how the XML works for editing. And now let me come to the Python. So Python steppables are where we're going to do most of the fancy part of our coding. And if you're a Python person, it's important to know their classes. But fundamentally, if you're not a Python person, the fact that it's a class is something that you can ignore. It's not, if the word class isn't meaningful to you, it's not going to be important, I don't think, at least not for a while. And as typical with object-oriented languages, fortunately, CompuCell isn't, CompuCell and Python aren't quite as verbose as, say, C++. Object-oriented C++, you get a lot of boilerplate header code before you can do anything. CompuCell tries to hide that from you. So you'll see from PySteppables import star, that imports essentially all the functionality of CompuCell. Import CompuCell, same thing. Import sys, And you're never going to change that. You might add something like, I believe that, for example, NumPy is loaded as NP, so NumPy functions are available now by default. So this may be a slightly old slide. Emma, question.
I write from the library import star instead of just import the library.
Okay. Perfectly reasonable question, and I do not know the answer to it.
I think I've seen, I mean, I would use from import when there's like one function or class I want, but I don't know, I haven't seen this one. Maybe there's something.
So Emma asks, why is it from Steppables import star rather than just import Steppables? And Python has a couple of different syntaxes for doing similar things. And I actually don't know whether there's any difference in terms of functionality, execution speed, in terms of which way you do those imports. It's a good question.
I think it's not, it's the same.
So Pedro says he thinks it's the same, in which case it's probably just historical that somebody liked it one way and somebody liked it the other. The boilerplate in old versions of CompuCell was quite a bit longer. There were a lot more initializations. Those are hidden now. And so that means that if you try to load code that was written 10 years ago, the core code will work, but those boilerplate initializations at the top of the files have to be edited to get it to work. Okay. Then you'll see a declaration class, and that will give you the name of the steppable. It will be the name here, single cell E599 L1 steppable. And then the inheritance pattern, steppable base pi. And again, if you're not a Python person, you can essentially ignore that and just understand that all of that stuff above those first 5 lines are boilerplate, and the code that you're going to be working with will be below that. And the Python cheat sheet we handed out covers these basic ideas. Again, the idea of steppables is that you move the cell, you update the configuration of the cell lattice, and then you call the Python. Then you update the configuration of the cell lattice, and then you call the Python. You have some freedom to adjust the frequency with which those alternations happen. But there isn't fancy cross communication during the 2 steps. There is a fundamental speed limit for movement in the cell lattice that you don't want cells to move more than about 1/5 of a voxel per Monte Carlo step. And if Pedro covers explicit forces, you'll be able to explore that in more detail. But you can call steppables more frequently than once per Monte Carlo step, or less frequently than once per Monte Carlo step, to try to synchronize. Okay. Within the steppable, there are a bunch of functions. Most of these functions are fairly self-explanatory. The one that is very Pythonic is the first one, which is the init, the double underscore init double underscore, and that has to be there in any Python class. And it essentially creates everything else that's going to be used in the simulation. Most of the time we don't mess with things around the init, but we're going to find that there are a couple of things that we do. If we want parameters to be available globally within the steppable, they go there. And tracking fields, which are a very powerful feature of CompuCell, go there as well. However, the remaining functions who have more explanatory names are things that we will use all the time. The start function is called once at the beginning of the simulation, and that's where we're going to put things like initial condition generation, setting up the values of parameters initially, and so on. The step function, as its name describes, is something that's called normally every Monte Carlo step, although you can adjust it to be every 2 Monte Carlo steps, every 5 Monte Carlo steps. And that's where the meat of the computation is going to be done. And then, depending on what you're doing with the simulation, if you want it to, at the end of the simulation, output, say, a file, there is a finish function, which is called when the simulation runs out of time. And there's also now an on-stop function, which is called when you hit that stop button in Player. One thing to remember, if you're really not a Python person, is that Python defines code blocks by indentation. And so you do have to be careful about indenting properly. Twedit has a block indent and block outdent buttons. So if you select a block of code and you hit indent in Twedit, it will indent correctly. But it's quite easy to get errors in Python by having indentation on. I think everyone who learns Python hates indentation as a code block definition. And then after a while you get used to it, you think, why does any other system exist? But that's one of the ones we have to do. Now, Twedit has lots of Python code snippets as well, maybe too many of them. And I really encourage you to walk through and pull down those menus and walk through those menu hierarchies. They're not always exactly, things are not always exactly where you might expect them to be. And there are still a few things that are missing, but most things that you want to do most of the time are there. And so if you say, gee, I don't remember how to reference a cell dictionary, it's there. There are a couple of things that you'd like I don't remember how to do periodic boundary conditions. That winds up not being in the Twedit code snippets. But a lot of the things that you would normally have to go to a manual to look up are in these code snippets. You will have to position your cursor in the right place for doing the things that you need to do. And Twedit code snippets will often tell you this goes in the start function, this goes in the init, this goes in the step function. Occasionally there are code snippets that actually do complicated things, and then you have to by hand move things, because sometimes part of the code snippet has to go in the start and part has to go somewhere else. But you're going to use some of these a lot. A cell attributes one is going to be used a lot. Extra fields automatic tracking is one you'll use a lot. Scientific plots is one that you'll use a lot. Secretion uptake field integrals is one you'll use a lot. And visit is another one you'll use a lot. The other ones we'll talk about, but those are the ones you really have to pay attention to. Twedit allows you to add steppables. In class, we will very often put everything into one big steppable. That's not really very Pythonic. It's not very modular. And so you could have a steppable that does the initialization of your code. You could have a steppable that does looking for cell death. You could have a steppable that looks for cell division. You could have a steppable that does analysis and output. So it's up to you to decide how to modularize your code. If you want to add a steppable, there is a button in that pull-down menu, Add Steppable, that will create the code that you need, the code template you need. And in particular, all the steppables need to be registered, which is what that main Python script does. And if you create your steppables using that function there, the updating of the main Python loop is handled automatically for you, which is a big help. Guess maybe I better close the door. Hold on a second. If they're going to be vacuuming, excuse me for a second. Close the door. I don't think I've ever seen them clean this building at all, so that's a novelty. Okay, sorry about that. So, one that we're going to use a lot is the cell attributes. And when we're talking about cells inside of Python, cells are complex objects. They have a variety of attributes, some of which are going to be very familiar to us already, some of which are going to be things we're going to talk about later on. Cell.id. Every cell has a unique number associated with it. An index. Cell.type. What's the type of the cell? Cell.volume. What's the current volume of the cell? Cell.target volume. Well, we've done that before with the XML. Cell.lambda volume. Cell.pressure. And cell.pressure has a little bit of a booby trap associated with it. Because if you define the target volume using the XML, then cell.pressure doesn't work. So that's something we should be careful about. And cell.surface tension, which is essentially the equivalent to pressure from the surface constraint. And you'll see if we look that there are a bunch of other cell attributes, cluster ID, which we'll get to later in the course, fluctuation amplitude. It's possible to actually have the amplitude of the fluctuations vary cell by cell, which which we haven't. Actually, I've never really explored that. It's an interesting concept. We could probably do some exercise on that. Cell.inertia tensor. What's the shape of the cell? And some others. Access modified dictionary entry is going to be one that you'll use a lot, although we haven't really talked about dictionaries yet. Okay. There are a couple of things that you might want to watch out for. Plug in the XML, remember you had to define the types of the cells. And you created a list which was cell type, and then there was type ID and type name equals. Cell.type returns that number. So if the cell were type dark, it would return the number 1. If you want to turn cell type into a name, you use self.typename in all caps. So capital D, capital A, capital R, capital K. And if you want to get the type by name, you have to use self.getType underscore name by cell of cell as a function. That's a historical issue. Originally, cell types were numbers, and so the naming was added later, and that's why it's a little bit awkward. Okay. Iterators. A lot of the time, we want to do something to all cells, or we want to do something to all cells of a given type, or we want to do something to all cells next to a cell. Anything where we're going to iterate. Python iterators are not quite the same as iterators in other languages, in the sense that the sequence of steps when you iterate over a loop with an iterator isn't necessarily guaranteed. Python iterates over lists. And again, if you're not a Python person, that distinction probably can be ignored safely. But the one thing you can't do is you can't change the list while you're iterating over it. That'll generate an error. And so, for example, if you're iterating over all cells and you delete a cell or create a cell, you caused a problem. So you'll see a lot of code where we stack something. We're going to make a list of all the cells that we might delete, or all the cells we're going to create. Then you get outside of that loop, and then you do the creation and destruction. Sometimes you're lucky, and changing the list that you're iterating over doesn't cause a problem. But Python, that's a Python no-no. So that's one you have to do. The classic Python syntax that we're going to use really all the time is for cell in self.cell list. and that's going to do something to all cells. We might print the volume of the cell. We might print the ID of the cell. Another one that we'll use a lot is for cell and self.cell list by type, where we'll do something to all cells of a given type. And there's some other iterators that you've got there. All lattice pixels, you typically want to avoid that because it's a very slow thing to do. Python iterators are not fast. All voxels next to a cell, all cells next to a cell, all voxels within a cell. So there are a lot of built-in iterators so that you don't have to remember how to do those. Okay. And again, if you're going to reference cells by type, the thing you have to remember is that the name of the type goes in all caps. So if you have cell type dark and cell type light, it'll be capital L, capital I, capital G, capital H, capital T. Now, another thing that we're going to have to face, and there's some inconsistencies in CompuCell dealing with this, again, historically, I don't think I would do it the way we did it these days, is that the XML predates the Python. And if you specify values in the XML, in general, you can't overwrite those with Python. Now there are some times where you can, but in general, you can't. And in particular, for the volume and surface constraints, if I specify the volume constraint the way we've been doing it in the XML, then I cannot change those at the single cell level. But you still need to call for the energy functional itself. And so if I want to change the cell volumes on a cell-by-cell basis, in my XML, I have to say plugin name equals volume, but nothing else. If I were going to do this today, what I would do is I would have the XML specify the initial values and then let the Python overwrite them. And the more recent plugins are written that way, but the old ones are not. So that's again, usually it's not a problem, but occasionally you can create bugs that are hard to track because you forget something like that, and you think it doesn't. If you try to overwrite a value in Python that you can't overwrite, it will not throw an error. It just won't do it. You'd rather have the code crash than to happily go ahead and not do what you thought you asked it to do. So if I'm going to specify volume and surface, which we've done repeatedly already in Python, those typically go in the start function. And suppose originally I want my cell target volume to be 25 and my cell lambda volume to be 2. If I were going to do that in the XML, I'd say plugin name volume energy parameters, cell type, and then lambda volume is 2, target volume is 25. The way I'll fix that is I'm going to now replace the plugin in the XML with just plugin name is volume. And then in my start function, I will go for cell and self.cell list, cell.target volume is 25, cell.lambda volume is 2. And so those are achieving the same thing. But now I have control at the individual cell level. And there are a variety of what are called scopes that one has to think about in a model of this kind. For example, all cells of a given type, or a specific cell. And you want to be able to do things to all of those classes, all of those groups. Okay. Something that's pretty important in CompuCell and gives you a lot of flexibility, but it may not be so familiar to people who are not Pythonic, one of the most powerful concepts that Python offers you is a Python dictionary. And so a dictionary is a place where you can store information, like a list or like an array, but it is keyed to a string name. And every cell comes prepackaged. In the old days, it wasn't. You had to add the dictionaries, but every cell includes a dictionary. And so I can add to a cell a key which I put in the dictionary. So if I want to create a new attribute for a cell, instead of having to do something fancy, I create the attribute by saying cell.dict, quote, whatever I want to call the attribute, equals whatever it is. And the nice thing about dictionaries is that they allow anything, any object class can be stored in a dictionary. So I could say cell.dict name equals, quote, hello. So in that case, my dictionary entry is a string. I could say cell.dict value equals 10.6, in which case my dictionary entry is a float. But I could also say cell.dict list history is 10, 11, 13, 15, which is a list. I could even put a dictionary into it. I could even put a class into it. So if I really wanted to put PyTorch, which is a class, into a CompuCell cell, I can say cell.dict my PyTorch equals PyTorch, and it'll work. I couldn't believe it worked, but it does. It's pretty slow, but it does work. And so the flexibility of dictionaries is tremendous. The ability to misuse dictionaries is also tremendous because of that flexibility. So we will use them in a gentle way, typically, but it's quite powerful. For example, if I have a list and I want to say, suppose I'm doing a cancer simulation, and I want to know the evolutionary history of my cells. So every time the cell divides, I can stack onto the list the history of divisions that I had. I could stack onto the list the exposure that I had to something. And so I could use these dictionaries to store quite complex information about the cells. You could say, well, why is it important to do it this way? Well, if you're building a complex simulation, I don't know a priori what kinds of information you're going to want to add to your cells. One of the things that we will add a lot are networks, complex biochemical networks to cells. The complex biochemical networks actually implemented C++, but the access to their values is through the dictionary. So you'll have cell.dict, essentially. It'll look a little bit different, but it'll be a dictionary entry, which will be the concentration of each chemical. So getting used to, again, syntactically, it's a little bit, it's a lot of characters to type. Instead of cell.x, you'd have to type cell.dict, quote, square bracket, quote, x, close quote. But once you get used to that, it's a very, very powerful way of storing information. I think that's one of the big strengths of CompuCell. There are a couple of things that I'll be honest when I think there are things that can be confusing or limiting. But there are some things that I think are quite powerful and flexible. And most of the other modeling packages don't have this level of flexibility, this idea of a dictionary attribute. Another thing that you may want to do, and this is something that's going to come up primarily if you use multiple steppables, is that variables are local to steppables, just the way when you write a function definition, the variables inside the function aren't accessible outside the function unless you pass them. And one way around this is to use globals. And you could global things in Python if you want to. But that's discouraged for a variety of reasons, and it's not very Pythonic. And so CompuCell comes with a dictionary, a global dictionary, and I don't know why Maciek didn't call it self.dict rather than this annoying name. but it's self.shared steppable vars is the dictionary, the global dictionary. And so that one you have to remember. I have to keep looking that one up because it really, to be orthogonal, it should be cell.dict and self.dict, but it's not. But remember, there is a global dictionary to store things on. You don't have to create it. And now I'd like to actually do some work, and I'm going to talk about cell growth and division. We're going to have to greatly shorten the discussion of cell cycle and the biology of cell division, which is worth a lot. You could spend, if you were taking a cell biology course, you'd probably spend a couple of weeks on the topic. But let's just talk about cell division at a very, very high level, at a CompuCell type level. One of the things that CompuCell does not implement is the cell cycle. PhysiCell, Paul's package, has the cell cycle built in as a concept, which I think is nice. Of course, biologically, there always are going to be some examples which do things a little bit differently. So, but there is a standard pattern of the way cells work. So in a multicellular organism, and people interested in the history of multicellularity will know why this is an interesting problem. Cells always have to make decisions. In particular, they have to decide whether they're going to proliferate, that is, grow and divide, survive, otherwise known as quiescence, which most of your cells are doing most of the time in your body, differentiate, that is, change their cell type, usually something more specific, sometimes the other way, or die. And cell death itself comes with many, many different ways of cell death, which have different consequences. And the decision whether you're going to grow, stay there, or die, or change your type, depends on a whole variety of signals. Some of those signals are the internal cell state. Some of those signals come from outside. And on the right, we have a schematic of some of those major signals. And just like the Russian novels, there are a lot of characters, and there are going to be a lot of genes. And in general, my advice about genes is that skim the list, and then if you see that the same character, so to speak, shows up a lot, then you might want to learn about that one. If you're dealing with inflammation and damage, NF kappa B there is going to be very important. If you're not interested in inflammation and damage, NF kappa B probably isn't going to be in your character list. There are marked here a variety of extracellular signals. Those signals can be received in different ways. They can be transduced in different ways. If you're Maria Bonderson, who's an expert on estrogen, estrogen has the unusual property that it enters through the cell membrane and then transduces the cell directly and not through a transmembrane receptor. Some of these transmembrane receptors bind diffusible signals. Some of them are contact signals to other cells. There are a whole variety of different kinds of signals. Some of them promote cell death. Some of them promote cell proliferation. Some of them promote changes of cell type. And where they're received and how they act varies a lot from signal to signal. Emma?
For a given cell, are those receptors binding like survival factors, death factors, and growth factors, all these things at the same time, and the cell essentially does what gets voted on by?
So Emma asks, there are a lot of different signals. Some are survival factors, some are growth factors, some are death factors. The cells are going to be receiving lots of different conflicting signals at the same time. Do the cell vote on these?
Like, yeah.
And the answer is effectively yes. Exactly, it may not be a simple majority vote because it may depend on the it'll depend on the internal state of the cell and its history. But in general, at any given time, the cell will be receiving signals promoting proliferation and inhibiting proliferation, promoting cell death and inhibiting cell death, promoting survival and inhibiting it, promoting differentiation and inhibiting it. And so the decisions have to be made by integrating that information. That information is integrated both through signaling networks and through gene regulatory networks. Gene regulatory networks typically operate on a slightly slower time scale than signaling networks. And then there also are metabolic networks, which we typically don't show here, but can have important effects that way. They're the things that produce the energy and the structural components of the cell. But they also are very heavily involved in cell death. And so the mitochondria, which are the cell's basically factories of energy, if the mitochondria are damaged or sense damage in the cell, they can do things that cause cell death, rather very rapid cell death. And so they're not actually really shown in this diagram at all, and they have their own set of complex metabolic pathways and cell death pathways. One thing that's labeled on this diagram, and I don't want to dig into it too much, we have cell proliferation as an output of what's happening in a very vague way, because we have just an arrow, a box called gene regulation, which is pointing to cell proliferation as if that was one thing. And then we have, on the outside, we have a variety of individual molecules that are pointing to a box called apoptosis. Apoptosis is one of the ways cells can die, by no means the only one. Apoptosis is the most orderly way of cell death. It's a form of cell suicide that has essentially the cell break itself down in an orderly way so its components can be reused. And particularly apoptosis tends to be non-inflammatory. Other kinds of cell death, like necrosis, is what happens when you really clobber the cell, turn off its energy factories, and it bursts. That's essentially a disorderly cell death. There are a variety of organized pro-inflammatory cell deaths like pyroptosis and necroptosis, which were only discovered fairly recently, which are involved in immune response. There are a couple of general principles here. You have to integrate these signals, which are going to conflict with each other. Things called growth factors often promote cell growth and proliferation. I guess that's not wildly surprising given the name, but names aren't always consistent. And you'll see lots of genes love abbreviations and acronyms. And I will try to remember at least the first time I mention a gene name to say the name out, but I won't. Sometimes I will forget. And so you will see genes that are things like IGF-1, TGF alpha, EGF. The GF in all of those stands for growth factor. Not every growth factor has a GF in its name, but if you see a GF in a name, it's probably growth factor. And exactly how they're transduced is variable. Well, we're not going to do a lot of network modeling in this class. We will talk about how to do it, and we'll show you how to do it, but we're not going to dig into that in huge detail here. Another thing that's something we're going to implement, maybe not today, given where we are in terms of time, but then immediately next week, is that there are a couple of basic principles of biological organization here. One of which is that cells have molecules that bind to the extracellular environment, into the extracellular matrix, the fiber architecture around the cell. And those molecules are primarily integrins, called integrins, although there are other ones. And the integrins, besides sticking, and I mentioned this the other day when I talked about cell adhesion molecules, most biological molecules have multiple functions simultaneously. And very often they do something physical or structural, and they send signals. And so integrins which bind cells, help cells stick to non-cellular structures like bone or cartilage or extracellular matrix, those integrins, if they recognize their target, in other words, if the integrin says, I am attached to the right kind of extracellular molecule, it will send a signal to the cell saying, survive. If, remember the basic principle that cells don't have roadmaps. If a cell integrin binds to the wrong target, then the cell is probably in the wrong place. And the way biology tends to handle these things, like viral infection, once a cell's in your body is infected with a virus, never is a long time, but almost never can that virus be eliminated. The only way to get rid of the virus is to kill the cell that's infected. And so similarly, in a lot of situations, if there's a signal to a cell that says something went wrong, the strategy that the cell follows is to kill itself via apoptosis. And so if the cell's integrins don't bind to their targets, the cells will essentially have a response, which is, I'm in the wrong place, something went wrong, it's probably me, therefore I kill myself. And so the integrins send survival factors if they're bound to the correct ligands, and they send pro-apoptotic factors if they're bound to their incorrect ligands. Similarly, these cell-cell contact molecules like cadherins, which are also responsible for cell-cell adhesion, transduce a signal that has a slightly different effect. Suppose that I have a tissue with a certain density of cells, which means my cells are all touching each other. The contact between the cells through the cadherins sends a signal saying the local density of cells is correct. That promotes cell survival and inhibits cell proliferation. If the cadherins are not bound to enough of their counterparts, that means the local density of cells is not high enough, which means there's been tissue damage. And that will then send a signal to say proliferate to fill in the gaps. And so these are both what you would call homeostatic mechanisms. If the cell's in the wrong place, get rid of it. If there aren't enough cells, create more. And the nice thing about mechanisms like this is that they can happen autonomously locally to maintain the global structure of the tissue. And so these are very powerful mechanisms, which we'll be able to investigate not quite today, but pretty quickly. And so it's useful to think about or to ask the question, what information is available to a cell that would allow it to build and maintain the very complex structures and functions that we have in our bodies? Again, the cells only have very local information. They know what the local chemical concentrations are. They know what they're in contact with physically. And there are some cells like neurons that have very long processes, so they can explore long distances in space. But most cells don't do that. Most cells have very local information. And yet your body is composed of these rather large structures that are functional and maintained. And so it's quite interesting to ask the question, how do you build functional and maintain complex large structures given essentially local information? And these kind of global principles of organization, like contact inhibition, proliferate if the density is too low, stop proliferating when the density is high enough. Or error correction, survive if I'm in touch with what I should be in touch with, get rid of myself if I'm touching the wrong thing, are things to think about. Okay, cell cycle. Again, CompuCell doesn't implement the phases of the cell cycle. If you've taken a basic biology course, you've probably seen these terms. If you haven't, we're not going to need them too much in the class. If you're going to do a cancer model, these will become more important, for example. The process by which cells grow and divide is pretty complicated. And the reason that it's so complicated is that if cell growth and division is not regulated properly, you're either going to have your tissues to degenerate, or you're going to get a tumor, or you'll at least get disorganization of the tissue. Most cells, most of the time, will be in G0, which is quiescent state, which is they're sitting there, they're happy, but they're not growing or dividing. Cells will exit G0 and go into cell cycle, and the cell cycle consists of a variety of steps. Those steps have to be executed correctly in order for cell division to happen properly. And certainly in later stages of things like cancer, you'll get disorganization of the cell cycle. You have G1, in which you have protein synthesis and the cell grows, the mass of the cell grows. You have S, in which the DNA of the cell is replicated. You have G2, where you produce additional proteins specifically needed for cell division. And then you have mitosis, which is actually how the cell divides. And there's this complex dance of the chromosomes and the nuclear structures during that. And then the actual cleavage and telophase of the cleavage of the cell into two. Normally, cells, after they've divided, will become quiescent, exit the cell cycle, and then have to re-enter it. There's some cases where that doesn't happen. And if you're familiar with development, there are all sorts of counterexamples to this basic pattern. In Drosophila, you have 10 rounds of nuclear division with no cleavage of the cells at all. So the cell goes from having essentially one nucleus to having 1,024 nuclei, and then all at once it falls apart into 1,024 separate cells. So actually the cleavage into separate cells and the nuclear replication can be separated from each other. Normally they're put together, but not always. And we think of cells as normally having one nucleus, but for example, in your liver, many of the hepatocytes in your liver have two nuclei in them as a normal state. So on the other hand, for example, within the cell division itself, prophase, metaphase, anaphase, if those aren't synchronized properly, the cell will die or else become cancerous. There really isn't a lot of choice. There are what are called cycle checkpoints. We're not going to implement those typically here so one concept, one engineering concept is the concept of a fail-safe. So you have to ask the question, if something goes wrong, what would you like to be done about it? And if you're driving a car, in general, cars have a pretty good fail-safe because if something happens to you and you take your foot off the accelerator, the car stops. Now, it may not stop fast enough, it may crash before that, but at least the car stops if you're not actively pushing on the gas. If you're in a locomotive and you don't do anything, the locomotive keeps going. And so locomotives then had to have what are called the dead man, which is to throttle the thing back off so that the person driving the train actually actively has to keep pushing on it to keep it going. But because the default failure mode of a locomotive is if the driver of the train dies, the train just keeps going until it crashes into something. At least with a car, if you have a heart attack, the car will stop unless your foot gets stuck on the gas. And so with cell proliferation, there are a series of checkpoints. Have I finished division properly? Am I ready to grow again? Have I synthesized all the proteins I need? Have I finished resynthesizing, copying my DNA? Have I organized the DNA for separating it physically? There are a lot more checkpoints than these, and in reality, we're going to ignore most of them, but we are going to have to make some decision. In CompuCell, cell division is not, in reality, mitosis phase takes some amount of time. It's fast compared to the other things, but it still takes half an hour to complete. In CompuCell, in most computational models, you either have one cell or you have two, and so that happens instantaneously. So cell division happens instantaneously. But you're going to have some rule that triggers cell division. And that would essentially correspond to that G2 checkpoint that we've got here. And so if we think about cell division, this is that M phase. You see the spindles forming the separation of the chromosomes and then the actual division of the cells. We don't have all that internal structure in our cells. We don't have chromosomes. We don't have asters. We don't have microtubules. So we're not going to be simulating all of that detail. And I think the key thing to notice is that the actual cleavage of the cells happens very fast once it's happening. Now, we also have to think about that G1 phase. How fast does the cell grow? And the growth rate of the cell is going to depend on a bunch of things. One of the key things is how much nutrient it has, because the cell is going to take up glucose or some other sugar and convert that to the material, the physical material that's going to build the cell. If you don't have the material to eat to synthesize new cell components, you can't grow. If you don't have any oxygen at all, you can't grow. You can do with low oxygen levels, but you need some. You have to also think about the amount of space. If the cell is physically confined, that often inhibits growth. There are some rules about rates of conversion. The metabolic processes in the cell take some amount of time, so converting glucose into, say, lipids takes some amount of time. You need energy to do this conversion, and so there's some energy usage also. There's efficiency. The cell's growth rate will be modulated by growth factors. It's going to be modulated, as I mentioned, by contact with other cells. The cell can have a memory that will tell it, in your state, your current state, grow faster or don't grow at all. And things like toxins can slow down or accelerate growth. So there are a bunch of factors that could influence growth rate. In our case, we're typically going to assume that the cell takes up somewhat of what we call a limiting nutrient, whatever that is, and that its growth rate is controlled by that one number. But that's certainly an oversimplification. So the simplest thing we can do in CompuCell when we have cells grow is to have the target volume of the cell increase in time. And we already ran a simulation very early on where we had used Wizard to build a simulation of cell growth. We didn't look at how it worked. But if we take our target volume and we increase the target volume, we changed it actually in the homework a little bit, increase the target volume by a certain amount per unit of time, we can have the cells grow at a constant rate. As a reminder, we always have to change that volume plug-in. So it's just the bare volume plug-in and then have the volumes defined in the Python. So typically we'd say the cells start out, say, with a volume of 25, and then at every time step, their target volume goes up by 0.01. In reality, for any useful model, that volume growth would be dependent on the nutrient state of the cell, the growth factors available to the cell, the pressure exerted on the cell, and so on. So let's do that. Well, we're at half past. Why don't we take a 5-minute break and come back at 6:40, and then we'll build a little cell growth simulation. You've already done this with Wizard, but we haven't done it and looked at what's inside of it. So we're going to go back and essentially redo that little Wizard simulation of cell growth, and then we're going to dig into what it means, okay? So let me walk you through what we're going to do, and maybe people have already done it, in which case we can go through it very quickly. If people have an issue, we'll walk through it a second time. So we're going to do a simple cell growth simulation. So we'll create a simulation domain, which would be 100 by 100 by 1. If you want it to be 100 and 200 by 200 by 1, it's all right. And we're going to create a single cell type. You could call it cell. And you're going to give it a contact energy. our initial geometry, and we're going to use the blob initializer to create a single cell to begin with. And we'll use the plugins of contact energy, volume local flex, surface local flex, center of mass, and the growth steppable, which are all selected on the tick boxes in the wizard. And then once you've saved the project, you'll have to go in and edit it, and you'll have to change the radius of the blob initializer to make the size of the blob to be 5, so you only get one cell. And then in the start function, you're going to initialize the initial target volume of the cells to be to 25, lambda volume to be 3. And the step function, you're going to make the cell growth rate 25 voxels divided by 500 Monte Carlo steps. It's useful to remember what the units of things are. In other words, if you say that the growth rate is 0.01 voxels per Monte Carlo step, that's okay. But if you want the cell to grow 25 voxels over 500 Monte Carlo steps, writing it out explicitly as 25 over 500 isn't a bad idea. And then you'll save and run the simulation and analyze the results. So if we're going to walk through that using Wizard, the first thing you're going to do is you can adjust the simulation properties here to be 100 by 100. We can make the lattice periodic if we like. I usually set the pixel copy range to 3, and we'll tick blob initializer here. Okay. And then we'll create a single cell type called cell. And then we'll skip the chemical fields as usual. And then in the cell properties menu, we're going to select contact energy, growth, volume, local flex, and center of mass. And then create it. Okay. Once we've done that, we can go in and we have to change the radius of our blob and the initial size of our cells to be 5. And then in the Python, we have to change the initial target volume of the cell to be 25, lambda volume to be 3. And then we'll adjust the growth rate to be 25 over 500. Okay. I'm not going to walk through it. You should get something that looks like this.
It's growing.
Oh, no, but it is.
Yes. Not super fast, but perceptible. I think
It is.
yeah,
Yeah.
I think it was working the first time. It was just slower than I expected.
Okay. Did people get something that looked more or less like this?
Mhm.
No. William, you got it? Alex, did that work for you?
Yes.
Great. Okay. Okay. So, sorry, that's so okay, let's try something else. We had a target surface set. And one of the things that we're going to run into is that the target surface has to be adjusted to track the target volume, which is a bit of an annoyance. That's something that Morpheus does automatically, which CompuCell doesn't do. So let's try making the target surface 20 and the lambda surface 3. Save that and run it and tell me what happens. How did the simulation change? And I should say, you have to add in the XML, you're going to have to add the target surface constraint. And so you want to remember how to do that using Twedit. And I can do that. I'll take a few minutes or two to try it yourself to see if you could figure out how to do it. Use the Twedit code snippets. And you'll add surface local flex is what you need to add. So you're going to have to edit two files. You're going to have to edit the XML file, and you're going to have to edit the steppable file. And the purpose of this little exercise is to figure out how to add a mechanism that you didn't put in the first time, right? We used wizard to define most of the mechanisms, but when we used wizard, we didn't add the surface constraint. Now we have to figure out how to add the surface constraint. And if people are stuck, please ask for help. And if you've got it done, give me a thumbs up or something so that I know what to do. Okay, Emma's okay. William, you got it? Okay. Okay, so we changed those two things. Did you get something that looked like that now? No. So this is showing us a problem that if the volume is changing, but the surface isn't changing, remember we spent a whole class trying to figure out what the appropriate relation between volume and surface should be. So there's an issue with that. So we're going to have to adjust this target surface area to keep its behavior reasonable when we change the target volume. And the way to do that is essentially to ask the question, we could do it with a square or we could do it with a circle. We're going to want to increase the target surface as we increase the target volume. But if we increase the target surface linearly with the target volume, the surface will get to be too big and you'll get a cell that gets floppy as it gets bigger. In reality, the surface should go like the square root of the volume, but you could try the other one. So try changing cell dot target surface in the step function to be 2 times, here I have pi as if pi were implemented, so you could make it just 2 times 3, and then the square root of cell dot target volume. And I hope square root is square root? Is that right? I'm trying to remember, is square root of intrinsic Python function, or is it going to have to be np dot square root?
I thought it was math dot square root, but now
I think it should be just square root.
I think square root is intrinsic Python function. We'll find out. I don't think pi is an intrinsic one though, so that I'm worried by seeing that.
Maybe we'll have like from NumPy import pi, and it works.
It's if you just make pi equal to 3, you're not going to have a problem here.
Yeah, it's a bit clear.
But or just say 2 times 3 times square root, be all right. But try that. Now, does that make is your cell still square? Does it now behave better?
I checked it. I think square root is actually in the math library.
Okay, so it's math dot square root.
So if you're in the Python Steppables file, if you just add the line beneath the imports that's import math, I think you could then do math.py and math.square root.
Okay.
I don't know how it integrates with this code, but
It should. So Emma says if you at the beginning of the Python, if at the header of your steppable you do import math, then square root math dot square root should work. I think NumPy is imported as NP by default, so you should be able to. The other way to do it, which would make life a lot easier, and I should change the slide, is just exponentiate cell dot target volume star to the 1/2. And then you don't have to worry about loading anything, okay? I'm not sure how this slide has been lurking here with a sand trap in it. It's not a new slide, so I don't know how we managed to get past that without noticing it in the past. But did people get that to work one way or the other? As I say, I would just use the exponential, and then you don't have to worry about loading math functions. As I say, NumPy is by default now loaded as NP, so that should be available too. But did somebody try that, and did the cell is the cell no longer square?
Right, it's not.
Okay, does anybody need help with that, or has everybody got it? William, did that, it worked? Okay. So now it should look like this. And you could change that ratio. You could make it, instead of 2 times 3, you could make it 2.5 times 3, and then your cell will be very floppy. You could make it 1.5 times 3, and then the cell would be more rigid. So you can, but at least you have to know that you have to adjust the target surface as you change the target volume. Okay. Now I'm going to talk about cell division. We've seen cell division in an earlier simulation. Again, cell division itself is a pretty complex process. But in terms of our simulation, we have to make a relatively simple decision. We either have 1 cell or 2 cells. And so we need to have some criterion for when a cell divides. And so what are some possible reasonable criteria we could use for a cell to divide? Anybody? Emma says how big it is. And that's usually what we're going to do. We're going to say that if the cell's actual volume is twice its original target volume, we're going to divide it. So we're going to have a threshold. In reality, cell division tends to be triggered in a little bit more complicated way, and it's not hard to load a model of this. There's a beautiful Tyson model of cell cycle. And in reality, what typically happens, and I think I discussed this a little bit at the end of last class in sort of our biological philosophy part, is that the cell will make a molecule, it'll make a cyclin, and then it'll stop making the cyclin. And as the cell gets bigger through synthesis, the concentration of the cyclin drops because the volume goes up and the amount of cyclin stays the same. And when the concentration of the cyclin goes below a threshold, then that triggers division. And so that means that in principle, if you reduce the amount of cyclin without having the cell grow, you could also trigger cell division. And so there are a whole bunch of situations in which growth and division are not done together. I mentioned the example of Drosophila, where the cell falls apart into 1,000 cells simultaneously. But in the early mammalian embryo as well, during what's called primary cleavage, you have very little protein synthesis, but you have synthesis of DNA and division, so that you have the egg, which is pretty big. The nucleus of the egg will replicate, separate. The egg will split into 2 cells, and those nuclei will replicate. Those cells will divide in 2, and so on. And so the volume of each cell goes down by a factor of 2 on each cell division. So you don't have the cells growing, but you do have nuclear replication. And it's also possible to have situations where the average size of the cell goes up slightly or down slightly on each cell division. So if we divide at a fixed volume, we're effectively saying that the volume of the cells doesn't change through the cell cycle, but after each iteration of the cell cycle. We could imagine that the cells divide when the target volume reaches some threshold rather than the actual volume. Or we could imagine that the cells divide when there's some concentration that reaches some threshold inside of it or goes down below a threshold. There are many possibilities. Or we could imagine that the cell just divides after a fixed amount of time, that there's some timer in the cell that divides it every so often. So there are a lot of possibilities. In our simulations, we're going to assume that we're triggering from a volume to begin with. Something that we do have to think about when we think about CompuCell cells is that we start out with one cell and we have now two cells. So we're going to need to create a new cell. And one of the things we're going to have to do to create a new cell is decide what its attributes are. And that includes where is it, and what kind of cell is it, and how big is it, and all of those things. And in the real world, you get a lot of that for free, but in computation, you don't get anything for free. You have to do it. And there's a little bit of a terminological problem because you have one cell to begin with, and then you have two cells. What's the nomenclature? In other words, if you start out with cell number 96, and cell number 96 divides into two, do you have cell number 99 and 997, or is one of them still 96, and which one is the 96? And so by default, biology often talks about daughter cells, where the original cell identity disappears. Computationally, we're going to keep one of our cells' identities the same and create a new cell. And so the cell whose identity doesn't change is going to be called the parent cell, and the new one is going to be called the daughter cell. And the first thing we have to decide is spatially which parts of the parent cell go to each of the offspring. Typically, we're going to define a cleavage plane and say that all of the voxels on one side of that plane belong to the parent cell, and all the voxels on the other side belong to the daughter. But we have to decide what the orientation of that plane is. And there are going to be a bunch of options that CompuCell will give you for that. Some of which happen biologically, well, all of which happen biologically under one situation or another. Are the two daughter cells the same size or not? Is the volume equally distributed among the cells? Again, in most cell division, it's what's called symmetrical division, but there are definitely plenty of biological situations in which you get one big cell and one tiny cell after division. They don't have to be symmetrical. And if it's the germ cells, you have meiosis rather than mitosis, where you get blebs, which aren't really cells that come off. You can get some complicated situations. And then you'd like to know how the attributes of the parent cell are inherited: target volume, target surface, and so on. And then is the daughter cell the same type as the parent? Does the parent cell stay the same type? Does the parent cell keep proliferating, or does it stop after division? And we don't have those things prepackaged. If we had computational cell lines, then all of those decisions, you'd say, okay, in this cell type, it does it this way, and then you'd have that available to you as a demo. CompuCell doesn't have that yet. So there are a lot of possibilities. Computationally, cell division in CompuCell is implemented with a special steppable called the mitosis steppable. And this is one of the few cases where you have to have a special steppable. And it comes packaged with functions that implement the things that we're going to do the most often with cell division. In particular, to create a new cell, to divide the voxels between the old cell and the new cell, and to inherit the properties of the parent cell. Now, some things are going to have to adjust by hand. The target volume and target surface are going to have to adjust by hand. Again, we're going to have cell division triggered by condition. That condition will typically be the volume of the cell being some target. That could be more complicated. One of the things that I mentioned already is that it's very tempting to go through the list of cells, find a cell that needs to divide, and say, divide the cell. And that violates a core Pythonic principle, which is you don't change the list of the things you're iterating over while you're iterating. And so you're going to almost always see a structure where we create a list where we're going to stack the things that we're going to divide, iterate over all cells, fill that list, and then iterate over that list. So let's talk about the mitosis steppable. The mitosis steppable is a class. There is going to be in the step function an empty dictionary called cells to divide. Then we're going to iterate over all cells in self.cellList, check for whatever condition we defined for the cell division condition. Here, if cell volume is bigger than 50, and if that's true, we're going to add that cell to our list, cells to divide. And if you're not a Python person, that's one of the Python functions you're going to have to get used to. If you want to add something to a list, you use the .append method. Then, when we've decided on all the cells that are going to divide, we iterate over the list of cells to divide. And now we're going to do the division. And we have a variety of cell division options. Self.divide cell random orientation. That cleavage plane could be in any direction. In the 4.4 version of CompuCell that we had earlier, that was a bug in that, so the random orientation was fixed, which was not good. There is self-to-divide orientation along a fixed direction, so you can have the cells divide always in the same direction if you want. And then self-to-divide along major axis. If the cell is elongated, you split it to make two more round cells. And self-divide along minor axis, if the cell is pancake-like, you actually split the pancake down the middle, like a bagel maybe. That doesn't happen as much biologically, but all of these things can happen from sometimes. Then there's a function, which is a very powerful function called update attributes. And update attributes essentially is where the properties of the parent and the child cell are changed after the division. And typically what we're going to do is, if we've had symmetrical division, we're going to assign half of the volume of the parent cell to each of the offspring. And so we'll say self.parent cell target volume divided by 2. Now what we do with the surface constraint is something that we haven't talked about yet, so we're going to have to think about that. The tracking function, which we had before, where we set the target surface to be a constant times the square root of the target volume, is not a bad thing to do. And then probably the most powerful one of the really nice features in CompuCell is this function self.clone parent to child. And that copies all of the attributes of the parent into the child. And one thing that's pretty neat about Python is it has a function called deep copy, which is used here, which is unfortunately slow, but cells don't divide that often. And even if you have introduced dictionary entries to the cell by hand, those will get copied. So that self.clone parent to child will copy all of the things that you've added to the cell automatically. And that's a very powerful feature. The one thing that we will find that doesn't get copied when you do this is links. Links will not get copied, but we haven't talked about links yet. But almost everything that you can imagine adding to the cell by hand in your Python will automatically get cloned. And then by default, just as a demo, we change the type of the cell under certain circumstances. That's not biological, that's just an illustration of what you could do. If we didn't divide the target volume by 2, we'd have 2 cells with the original target volume, and the thing would grow very, very fast, because we basically, every time we divided a cell, the amount of volume of the system would double. If you tick the mitosis box in Wizard, you'll get the mitosis steppable put in automatically. If you are doing it from your existing simulation, and I'd like people to do this, if you select on the Python in the Twedit editor in the left-hand column and right-click on it, there'll be an option Add Steppable. And if you select that Add Steppable, you have to give the steppable a name. You could call it mitosis or my mitosis. And then there'll be a call frequency. Do you call it every Monte Carlo step, every 10 Monte Carlo step? And then a steppable type. And there are a couple of options here. Generic, which would be a regular steppable. Or if you're going to want to implement mitosis, you have to click mitosis. And there's also an option called cluster mitosis, which is special, which we're going to get to later on. Our cells at the moment don't have any internal features. They're just one big blob of material. CompuCell allows you to build structured cells that have subcompartments. And if you want those to divide, the rules for that division are rather complicated, and there's a special steppable that handles them. And the reason it's called out as a special thing is that it's pretty slow. And so you don't want to have to implement all those features unless you're using them. And you also have the option of adding extra visualization fields at the time you create the steppable. Okay. You can also add steppables through that CC3D Python pull-down menu with the same dialog box. Now, I mentioned already, I think this slide may be a little bit out of order, that if you have multiple steppables, variables created in one steppable are not available in the others. And so remember to use that self.steppable shared variables dictionary as a way of passing information between steppables if you need to. Okay. So if we want the step cells to divide when they've reached a predefined doubling volume, we say, okay, we create our mitosis steppable. We then create our empty list, cells to divide equals empty list, paired square brackets, for cell and self.cell list. If cell.volume is greater than 50, cells to divide dot append cell. And that will divide any cell that has a volume of 50. Again, once we've created our list of cells to divide, we have to run through that list second time. We're going to pick one of the cell division options, random, major, or minor axis. By default, we will assign the volume equally to the two daughters, so we divide the volume by 2. And then we clone parent to child so that the daughter cells have the same properties as the parent. So here's a picture. Before mitosis, we have a single object representing a cell. After mitosis, we have 2 objects representing 2 cells occupying the same voxels that we started out with. Self.parent cell will have the ID of the parent, so it represents a continuation of the old cell. Self.child cell is a new cell object with a new ID. So why don't you try adding to your simulation cell division? And here I said create a different simulation domain. Just leave the simulation size the same. Don't change the simulation size. But do add something. Add a mitosis steppable where your cells divide when they get to a target volume of 50. When they get to a volume of 50, half of the volume of the parent goes to each child, and the properties of the children are identical to the parent. Okay? And feel free to ask any questions if you have problems with that. I can help out for people who are online, and Pedro can help out for people in the room. And give me a thumbs up when you got it working. And I see the slide asks you to do some plotting, and I apologize. We rejigged the order of the slides a little bit so that we haven't covered plots yet. Well, that's the next thing we're going to do. Emma, you got it working? William? Still working on it? Alex, did that work for you? No?
Maybe.
Maybe. So, if you need help, speak up. Pedro, I don't know if you want to look over William's shoulder and see if. So again, you're going to create a new steppable called mitosis steppable. And then in mitosis steppable, you're going to set the condition for division, Alex. And then you have to say update attributes, parent.target volume divide by 2, clone parent to child. And if that, when you paste it in that mitosis steppable, if it did something like change the cell type, delete any text that has to do with changing cell type. And Alex, if you want to screen share and look at your code, we can do that too.
I can show you what it looks like. I'm just not sure.
Sorry?
I can show you what it looks like. I'm just not sure if it's right or not.
Okay. Well, if you run it, what happens?
It does make different cells, but they're all, well, I guess it's the, yeah.
Okay, let's take a look.
So it looks like.
That looks interesting. I wonder what the green is.
Oh, there's another cell type that I had. Maybe that's what it is.
Okay, let's look at your Twedit and see what you got there. It's a little fancier simulation than the one I was expecting, but there's nothing wrong with that.
It's because I added
Okay. That looks fine. So lines 71 through 75 are changing the cell types.
Mhm.
So why don't you comment those out to begin with? Another Twedit feature that's convenient is there's a block comment. If you select a bunch of lines and then you go under edit and hit block comment, it'll comment everything at once. Try that. Okay, why don't you save that and try running that and see what happens.
Yeah, this is a general condition, so.
I'm actually a little surprised that didn't crash, because normally if you change cell types to a non-declared cell type, it'll crash. Well, let's see what happens if you run it.
Now it's this.
Okay. Yeah, that looks pretty much like what we'd expect. Okay. Anybody else want to go have problems? Want to go over that? William, how's that going? Okay, everybody got it. Okay, so again, here I'm going to skip that for the moment. Here I created two cell types. and had the cell types change their type. So it was a little bit of a different exercise, but for the moment, that's okay. Okay, the last thing I'd like to do tonight is something that I'm never quite sure when to talk about scientific plots. Really, the earlier you learn how to do scientific plots, the better, because it's hard to do any analysis of your simulations until you can plot things. On the other hand, until we know what Python steppables are, we can't do scientific plots. And if we start out with all the Python for steppables, then we spend a lot of time on coding before we do anything modeling. So it's a little bit of an Ouroboros where everything requires everything else. So we're doing plots now. So I would like to talk about scientific plots. CompuCell has not an infinite range of scientific plots, but a reasonably flexible range. And everybody says, why not just use matplotlib? And the answer is don't ask that question. It has to do with library compatibilities and licensing issues. So we use a different package, which has somewhat similar functions to matplotlib, but isn't identical. And I will tell you up front that that package has some issues. And so that you may run into occasional fancy features of the package that crash. They're not ones that we will need, but I'll show you later or something that there's some things that you might be tempted to do that don't work. Okay, so if we're going to create a plot, we have to think about actually some computing now. Player gives you a display screen, and within that display screen, you see windows that represent your cells, and possibly we saw chemical fields and other kinds of things. And so if we're going to create a plot, the first thing we have to do is create a plot window in which we are going to display the plot, which will live inside of player. And that creation of the plot window is usually done in the start function. Then we have to create a data series, the things we are going to plot in that plot window. And that also typically goes in the start function. And then once we've created the plot series, we have to add points to the plot. And that's typically done in the step function. So there are three things we have to do. So we can have multiple plot windows. We could create 3 plot windows. Within each plot window, we could create multiple data series. And then within each data series, we add points as we need. And to help us remember that, Twedit has a set of functions under scientific plots. We have setup, which creates the plot window. And you'll notice it says start function. It's telling you where it goes. And then we have add data points, and it says that goes in the step function. And then there are a couple of things that we might want to do. We don't have to do our update anymore. The old prompts you saw you had to update by hand. We might want to erase a plot. One thing we'll find is that when we keep adding points to plots, if we have lots and lots of points, it actually slows the simulation down. Thousands of points is no problem, but if you're tracking cell trajectories in 3D, you could have millions of points in a plot, and that actually becomes slow. So you might want to erase it. And then you can actually export those plots as images or data from the Python. So you'll find that those code snippets are useful, but they do require a little bit of editing. So to create a plot, the first thing you're going to do is create a plot window. So you're going to position your code in the start function. You'll pull down that setup function, and that'll create a plot window, and it'll also create some data series, dummy data series. And the default code is going to look like this. There's going to be self.plot window. So this is now a handle to the plot window. And there's a function add new plot window. And the plot window gets a title. And the x-axis gets a title, the y-axis gets a title. And then you can make the x and y scales linear or logarithmic. You can have grid lines, and there are a variety of other options, which I'm not going to show you at the moment. One thing that's a little bit weird is that the name of the title of your plot window has to be unique. You can't have two plots with the same title. Once we've created that plot window, there's a function add plot, which adds a data series to it. We can have as many data series in one plot window as we want. And so here we have self.plotWin.addPlot, and then here M volume is the name of one data series. And we say, is it dots or lines, and what color is it, and how big are the symbols in it. We could create as many plot windows as we want. We could say self.plotWindow1 equals self.addPlot blah, self.plotWindow2 equals a self dot, and so on. And so there are a variety of options here. Again, the data series within a plot window have to have a unique name. The plot windows themselves have to have unique names. And even if the plot windows have different handles, the title of the plot has to be different. I don't know why that is. You can have legends for your data series. When you create the plot window, there's a variety of configuration options. Those are a bit hard to remember. Configuration options, legend : true is one that you might want. And so that one will then have a floating legend that helps you remember what color is what in your plot. Okay. Once we've created our plot window to add the data series, we'll go into the step function. And if we pull down the add data points, by default, it'll give us two data series. Sometimes we'll want one, sometimes we'll want three or more. The data series, the plot window handle and the data series handle have to be the same, identical. Otherwise, it won't throw an error. It won't plot anything. It won't throw an error. And so here's self.plot window. That's where we're going to plot. Add data point, quote M volume, the data series name, and then the X and Y coordinates. Now, it's pretty common that the X coordinate is the time and the Y coordinate is some scalar value, but they don't have to be. We could plot cell volume versus cell surface. The X and the Y, X coordinate doesn't have to be time. But the handle, plot win, and the name of the series, mvol, here have to match exactly what you defined in the start function. And so typically here we could go through the cell list and plot the volume of every cell versus time. And of course, as we have more cells, we'll get more and more dots in that at each time. We might want to calculate the average volume. We'd have to do that by hand and then plot that. Okay. So why don't people try to do that as an exercise? Why don't you add to your step function in your simulation a plot which plots the average volume and surface area of your cells. And before that, let's actually, I skipped an exercise here. Before you do that, why don't you actually first plot the volume and surface area of each cell during the growth? Okay. And so that means that at the first time steps, when you only have one cell, there'll be one dot. When the cells divide, there'll be two, then four, then eight. So you'll see that you'll get lots and lots of points after a while. But let's do that. And that's the code that I have here up on the screen. And that's more or less the default code that you're going to get from Twedit. And why don't people see if they can get that to work first? And then once you've got that working, you can write a little loop that calculates the average volume of each cell and the average surface of the cells, and then plots that as a data series. If you wanted, you could create a new plot window and put it up in that, not get rid of the old one. Okay, is that a clear exercise? That makes sense? And again, I encourage you to use the Twedit code snippets for scientific plots. The basic one that you have up here on the screen is essentially, I think, exactly what the code snippets will paste in for you. Something that's in the slide deck that I'm not going to have time to cover today is that you can also plot histograms. So those are supported automatically. And if you're interested in that, for example, suppose you wanted to plot a histogram of the cell volume instead of the individual cell volumes. That's supported automatically in CompuCell. It's not hard to do, but that's one I may send you to the manual or to the slide deck for, rather than to have to do in class.
Thanks.
Okay, so one point that I should make here that Emma ran into that was, I think it was implied with what I said earlier, but it was not clear, is that the handle self.plotWin is a variable. And so it is local to the class in which you instantiate it. And so if you create the plot window inside of your growth steppable, then you have to add the data series to that plot window inside of the growth steppable. If you create the plot window inside of the mitosis steppable, then you have to add the data series inside of the mitosis steppable. Now, in principle, if you really want to do something fancy, you could pass the handle to the plot window through the global dictionary and then read it into the other steppable. But in general, it's easier to create the plot and add points to the plot in the same steppable rather than to try to pass it back and forth. So I actually thank you, Emma, for making that small error, because it reminded me to remind people of that. If everything is in one big steppable, then you don't have to worry about that. But if you've got multiple steppables, it's not unusual to have an issue where you create the plot in one steppable and then you try to write to it from another one. So really for this basic one, you don't have to write any Python at all, because the Twedit code snippets are exactly what you need. You don't have to change them. The only thing you have to do is put them in the right place. So in the start function, you don't want to put it inside of a loop, for example. And in the step function, you want to put it inside that 4-cell loop. And then for the people who've gotten it, they make it a little stretch exercise. Calculate the mean volume and surface area of the cells at any given time, and then plot that data series in rather than all of the cells independently. And also, if people have already got that, you can play changing the colors, changing line versus point, changing sizes of the symbols, and you also can try log scaling things or auto scaling. And if you're really ambitious, you can go into the manual and look up some of the options for the plot window. Again, I think the first time one does this, it takes a little bit of learning. Once you've done it a few times, it's pretty convenient because the concept is fairly straightforward. You create a window to display the plot, you create the data series in the window, then you add points to the data series. And so that set of steps is going to be the same every time. Emma, did it work? Did it work for you? Did it work for you now? So what you should see here for this first one is you'll see 1 point for the time before the first division. You'll see 2 points, then 4 points, and then 8 points. Now it may be that the actual volumes are identical, in which case they'll be on top of each other and you won't see it. Or maybe you don't have the add data points inside of a 4-cell loop, and so you're only plotting it for 1 cell. That's also possible.
No, I think before I did have them within 4 cells, but
Okay.
when I took them
Okay.
collected the cell
Right,
values.
right. Right. That's fine. Okay. William, is that working for you? Okay. Pedro, can you look over William's shoulder for a second? And Alex, if you want a screen share, I can try to see try to help you out with it.
Yeah. I think you explained this, but the
Okay, so let's look at your code. All right, so let's look at where you create the plot window. So the first thing, can we scroll up a little bit? Okay, so you have to create the plot window inside of the start function. Because here you're creating a new plot window every step. So you're trying to create the same window again and again and again. So if you grab lines 42 through 49 and move them up into that start, you don't have a start function. That's why. So, okay, so your growth steppable doesn't have a start function. That's why things are confusing. So if you type the words def
where should
in line there, def space start parenthesis self comma MCS colon close quote colon. Now hit return. Okay, now paste that material there. Okay, good. Now try running it. Okay. Start function requires one positional argument MCS. Okay. What did I do wrong? Can we scroll down to the mitosis step? There's no start function here.
One.
How about going up all the way? Oh, so yeah, sorry. So just get rid of this. Get rid of the MCS. Look at line 11. That's what we needed. Yeah. Okay. Fine. Okay. There we go. All right. That doesn't look too bad. Okay. Okay.
The exercise.
Yeah, I apologize. It, if it says, right, the instructions are to put it in the start function, but there was no start function. So that was a little bit, a little bit confusing.
And it crashed.
Sorry, crash now?
Yeah, I tried to stop.
Okay,
It
let's
crashed.
see what happened.
I'll even show you what it there now. It's just when I tried stopping it,
Oh, that's not a Python error. That's a, all right. What version of Compusel are you running?
Yeah.
the newest one.
So they grow.
Huh. All right.
And this is 2 years.
Well, I apologize for that one. That one I haven't seen before.
The bottom of each cell. There's here inside of the
It's fine now.
all cells can be encapsulated.
Or it's
Okay.
So the exercise is basically
All right. Anybody else have an issue or want to talk?
each cell calculates the mean on, and then it
I have
plots just
something that I'm
one.
curious about.
Okay, sure. Let's see.
So I
These
tried
two
to,
lines.
Here, why don't you just screen share it?
Second form. Calculates the mean volume. Just like mean volume plus equals.
Might as well if you, if it's interesting, let's see what maybe ever.
Okay.
Okay, great.
to do is figure out how to make two independent plots.
Yep,
So I thought, well, here's the like plot object.
yep,
So we're just making a new object.
right.
I set the title to what I wanted so I could.
Yes,
export it.
yep.
And then I go into that object and I append or like add these plots.
Yep,
So
yep.
this object is supposed to be for the mean values.
yep.
And I set it to have line.
Okay, yes,
Is this the right syntax for line?
yes,
Okay. Then I make a new plot and that's gonna be the individual volume values.
yep, that looks fine.
So I want dots for those.
Yep, that's fine.
But then when I went into the player, can you see the player? No.
Oh, you have to reshare for player.
I expect this one to have dots because it's the individual values and this one to have lines, but these are dots.
I can't see the,
Let me see if I can share the whole screen because I thought those windows would just be associated with the player.
yeah, you probably have to share the desktop. Because this is one of these things where if you're having an issue with it, almost certainly everybody else is too. So it's better to share it together. Okay.
So here's what I'm seeing.
Yep.
This is the plot that I should be writing for the new
Right.
and it's supposed to have lines, but
Right.
it's dots.
Okay. And the other one?
And the other one is plain.
Okay, that means that somewhere our handle name is wrong. So there's probably some subtle typo, like we've got a capital letter or plot win 1.
Oh, I think I know what it is. That's.
There we go. Yep. Okay.
Yeah, I think this will fix it. Oh. Did I not see it? So this is window 1, new. Window 2 is individual.
Yeah.
Window 1, well, this is individual. Yeah. And
Yeah.
window 1 is me. So I think I'm adding them to the right places.
Yeah, that looks right.
Maybe it just didn't, I mean.
So one thing you might want to try, should be okay, but make the data series names different. Make one mvol and one mvol2. It's just like the title of the plot. If you've got a different handle, it shouldn't care because it should reference the handle, but sometimes it seems to not like having a repetition.
Yeah, that's probably that.
Oh, I know what the other one is. You've got vol and sir rather than mvol. Okay, you got it now? Okay, go. Okay, try it.
Like, yeah, I just changed that.
Okay, fine. Yeah, let's see if that does it. So you don't have the duplication.
And now they're both the same. So okay, let me check to see whether the mean is well.
But you've got multiple points, so in other words, at each time you should only be plotting 1 point. So because you're not inside the loop. So the mean volume one is not doing what we expect, huh?
Right, the individual one is working. The strange thing is that the mean volume one was working before I decided to add the individual, like the summarize. It was just the line.
Plot win 2. Plot win 1. Looks right.
So what I do is select the volumes and surfaces. Then I just do empty stack to make it into.
That looks fine.
Then I just don't put. Oh, I suppose. No, this shouldn't be the way. This should just be the. But let me. Actually, maybe
It is a number you do and you don't need.
Not a list of all the values.
it might be one number within an array. So
Yeah, if so. There we go. So in other words, you were creating a list which had all the values in it. So
if it's an array.
yeah, I've never tried to plot an array like that. So no.
I just forgot because it was only one number in the array, but I think that NP doesn't know. No, I think that it did like this. This was not correct.
So it's volume is a scalar.
But I have to go, otherwise I lose the bus.
Uh-oh.
So,
Okay. Well, thank you. We're going to. Now they're doing this. Huh.
so it's printing in the terminal, not there. So the terminal.
Oh, it's in the.
William, how about you? How's it?
I got it.
You've got it.
But I mean.
Except it's not.
But
not
I think I got it.
Yeah, it's not.
Wait, oh no, these are 10 steps. So actually, that is.
You're printing inside the loop. Go
Yes.
to the
So I'm printing within one step. So every time there's a step, it should print. So it doesn't.
Yeah, every step, but not inside the loop, so it's correct.
Okay.
It should be correct, but it also should be like a rational number, because you're doing a mean. Doesn't make sense to be like integers.
Okay.
There's something wrong. I mean, I will do the simple one, like just sum all the volumes inside the loop and divide by len.
Yeah.
Solve these. That's what.
Got a quick question.
Yep.
For my homework today, because
Sure.
my computer cell is, I don't know why it doesn't work from last week till today. And I work with Pedro
Mhm.
like before the class and we re-download it and stuff. So if it's possible, I can extend the
Sure.
videos for that.
If you get to me by Friday, that's fine.
Okay, yeah, I'll do that. Thank
And
you.
if you're having trouble with things, okay, I think for today that's probably people play. I realize that figuring out plots is not absolutely trivial. It looks like there may be some issues. I'm going to let people go. I'm going to try to help Emma see if I can figure out what's going on with hers. And I hope people can take this and play with it a little bit and explore. And next week we'll continue with plots and move on from there. Okay? Thanks, everybody. Good night.